I'm Dr. Momita Bandopadhyay, Assistant Secretary of the Archana Sharma Foundation of Calcutta and also a faculty at the Department of Botany, University of Calcutta. So on behalf of the Archana Sharma Foundation of Calcutta and the Department of Botany, University of Calcutta, I welcome you all to this national online seminar on the occasion of the 4th Professor Archana Sharma and Professor A.K. Sharma Oration. And we are very happy to welcome all of you. And we hope you will be exposed to a very stimulating oration by none other than Professor RAJEEV VARSHNEY, who is I don't think he needs introduction, but anyways, so Professor Varshney will be speaking on he's one of the very famous plant scientists of India, and he will be speaking on one of the issues that is really a very important issue in the context of present day and for food security of our country and also of the world. So he'll be talking about genomic interactions for crop improvement. And we have a program schedule, which I think all of you have from the flyer that we circulated. So the next order of business is actually opening remarks of Professor Anita Mukherjee, who is the Vice President of our foundation. So over to you, ma'am.
Thank you, Momita, very much. So on behalf of the Archana Sharma Foundation of Calcutta, I welcome you all and wish you a very good morning. I know we are all eagerly waiting for the 4th Archana Sharma and A.K. Sharma oration to be delivered by Professor Varshney. But please permit me to say a few words about the foundation. The Archana Sharma Foundation of Calcutta was established in the year 2009 by Professor Arun Kumar Sharma and his associates in memory of late Professor Archana Sharma, who was also a great cytologist as well as his life partner in all true sciences. The foundation is registered under West Bengal Act of Society and is a non-profit organization. The principal activity of the foundation is to publish out the journal Nucleus, an international journal of cytology and allied topics, published by Springer Nature, India. The royalty earned every year from the publication is utilized in social welfare activities concerning education of meritorious underprivileged students, particularly the girl students of our society, to pursue higher education. The other activities of the foundation, besides conducting annual memorial orations like the present one, includes organizing lectures, seminars, and symposia, singly or in collaboration with the Department of Botany, University of Calcutta, and other scientific bodies and societies. The foundation has collaborated in 3 international symposia since 2015, which were held at IICB Kolkata in the year 2015, 2017, and 2019, which were held at the IICB Kolkata. Very soon, we are going to host the website of the foundation, where you will be able to see the past and present activities of the foundation. We sincerely seek your patronage to accomplishing our mission and carrying out the legacy of the great teachers, Professor Arun Kumar Sharma and Archana Sharma, through the activities of the foundation. With these few words, I thank you all for giving your time and request Professor Chaube, the head from the Department of Botany, University of Calcutta, to take over this virtual stage of webinar. Thank you very much.
Thank you very much, Professor Mukherjee. And good morning, ladies and gentlemen. I can see a lot of new unknown names here. I will not say the faces because most of them are represented here by names. I hope that you are all fine. You are safe and taking good care of yourself and your family members during this pandemic. We know that the COVID-19 has affected directly or indirectly almost every sphere of life in the last month. Some of the sectors are affected severely and some are little. And in the universities, particularly the ones which are non-residential, like ours, which are severely paralyzed, particularly the research activities. Even though we are somehow carrying on the teaching activities, but the research has been severely paralyzed. And particularly some of the campuses, universities which are residential in nature, they are somehow carrying out the, some research and academic activities, but ours is fully compromised, I can say. Under these conditions, when the academic is on the downside due to this pandemic, the small webinars, these activities of online, these online seminars or webinars, which are organized by our department or other members of the department, of different departments. It surely brings a smile on our face and gives us the hope that in coming time, obviously, it will be normal again. We will again see a normal future, normal time, and we will bring back our happy day once again. We will again move in the department, and we will not meet on the computer like this, but we will see our speakers face to face in our Archana Sharma Memorial Hall. So I welcome you all here, all the participants. I welcome our speaker of the day, Professor RAJEEV, VARSHNEY, all the faculty members who are present here, and all the students, the scholars who are joining in this webinar. I welcome you all, and I congratulate Archana Sharma Foundation once again for organizing this meeting, particularly during this pandemic period, to keeping the host alive, that we are still there with our activities. So once again, welcome you all, and I wish a great success to this seminar. Thank you. And I hand over the platform to Dr. Momita.
Thank you, Professor Chaube. We really appreciate your support and your kind words and your welcome address. So now I will request Professor Amita Pal, President of the Archana Sharma Foundation of Calcutta, to introduce the speaker of the day.
It is my proud privilege to introduce Professor RAJEEV VARSHNEY, an agriculture research scientist specializing in genomics and molecular breeding with more than 20 years of service in international agriculture. He did his PhD in agricultural botany from Chaudhuri Charan Singh University, Meerut, and then he did his postdoc in IPK Gittersleben, Germany for 5 years as Humboldt Fellow. He joined the International Crop Research Institute for the Semi-Arid Crops, Semi-Arid Tropics, that is ICRISAT, Hyderabad in the year 2005, and currently servicing as Global Research Program Director, Genetic Gens at ICRISAT. He is an either honorary or adjunct professor in 10 universities and institutes in Australia, China, Ghana, and India, including Murdoch University, the University of Western Australia, the University of Queensland, Australia. He is globally recognized leader for his work on genome sequencing, genomic-assisted breeding, and traditional genomics in legumes and cereal crops, and capacity building in developing countries as well. Professor VARSHNEY is a J.C. Bose National Fellow, and for his outstanding and seminal contributions, Professor VARSHNEY has been honored with several fellowships, awards, and honorary positions at international level. For instance, Professor VARSHNEY is an elected fellow of all the 4 science academies of India, that is Indian National Science Academy, National Academy of Sciences India, Indian Academy of Sciences Bangalore, and National Agricultural Academies of Sciences. Also, he has a fellow of several foreign science academies, including the German National Science Academy, the World Academy of Sciences, American Association for the Advancement of Science, Crop Science Society of America, and American Society of Agronomy. He is an honorary fellow of Indian Society of Pulse and Research Development and Arid Zone Research Association of India. He is recipient of several prestigious awards, including Shanti Swarup Bhatnagar Award in Science and Rafi Ahmed Kidwai Award in Agriculture from the Government of India. Professor Rajeev is recognized as a leader in genome sequencing, genomic assisted breeding, translational genomics, and capacity building in international agriculture. He has made a pioneering contribution by integrating advanced discoveries in genomics with crop improvement, especially in legumes in developing countries. Among different noted contributions, he has genome sequence of 10 crops, including pigeon pea, chickpea, groundnut, and pearl millet, and several molecular breeding products in chickpea and groundnut to his credit. Genomic resources developed by his colleagues and collaborators are being used in several breeding programs with an objective to modernize crop improvement in developing countries. He has provided strategic leadership as a principal investigator to tropical legume projects that have facilitated the release of 266 improved legume species and production of about almost 50,000 tons of certified seeds with the benefit potential of 5 million hectares and over 25 million smallholder farmers in 15 countries in sub-Saharan Africa and South Asia. He has published more than 450 papers in high-impact journals, including 15 papers in Nature Science journal. He is the first Indian agriculture scientist or plant biologist with an H index more than 105 and over 42,000 citations as per April 2021 report. Thomson Reuters Clarivate Analytics has recognized him as a highly cited researcher consecutively for 8 years, starting from the day 2014. And Times of India announced him as one of 10 most influential Indian scientists. He is also recipient of other prestigious awards. I am not going to read out all those just because we are eagerly waiting for his talk. So without much ado, I am handing over to Professor Rajeev VARSHNEY for his talk. Please, Dr. Rajeev, unmute yourself.
Yes. Good morning once again to everyone, and thank you very much, Professor Amita Pal ma'am, for very elaborative introduction. I really feel very privileged to deliver this fourth Professor Archana Sharma and Professor A.K. Sharma oration. And before that, I would like to recognize Professor Anita Mukherjee, Vice President of the Foundation, also Professor Vinay Chaube, Head of Department, who gave these remarks. And I'm really grateful to Professor Amita Pal for her invitation to deliver this oration. And also very good to see Dr. Momita for moderating the session. I have seen many senior colleagues, eminent scientists, well-wishers and friends in this session. I can see more than 225 people, and it's always pleasure to have such a great audience. And I would not like to mention all the names, but really good to see Professor Sopan Datta, Professor Sampadas, Professor Lavanya, and many other friends. So it's really pleasure to be here. As Madam has mentioned about that presentation, let me open my presentation first, and then I will get back to that one in a minute. Okay, so this is, I think I am having that privilege to share my window. If not, then okay, maybe this one I should do this one. No, okay, good. Thank you. So I hope that you can see my slides, or let me do this thing. No, there's some problem here. Can you see my slides, please?
Yes.
Yes,
Okay.
we
Yes,
can,
we
Professor
can.
Varshni.
Good. So thank you very much once again for this invitation. And this is really my pleasure to be here and to give this. I would like to close this one. Okay. Yeah. So this oration, and although I know these names, Professor Archana Sharma and Professor A.K. Sharma, since I joined my PhD program with Professor P.K. Gupta at Meerut University, I used to hear a lot and read about them, but never had an opportunity to meet them. But nevertheless, before I start my presentation, I would like to pay my sincere tribute to these two legends who are attributed with laying the foundation of science, education, and research in the post-independence India. And as we all know that these names do not need any introduction, but recently I was reading about them and I was so much impressed to note their contribution. So for instance, Professor Archana Sharma has been a renowned Indian botanist and cytogeneticist, cytotoxicologist. She has played a very important role in that education, not only in the research, but education as well. Published many nice papers, including in Nature, and was awarded with that Padma Bhushan by Government of India. And same thing for Professor A.K. Sharma, who is considered as a doyen of botany in general and cytogenetics and cytotaxonomy in particular. And he has made significant contribution to plant biology, especially in the cytogenetics, published more than 600 papers, including 6 in Nature, and also again received large number of awards or so. So I think it's really an honor and privilege for me to deliver this oration in their name. And they have left so many legacies, but among those things, I think 2 are very important. One is this reference book for chromosome staining. I know that this book always was there when I was with Professor P.K. Gupta at Meerut. And I know the journal, the Nucleus Journal, which started way back in 1958, and now this is being published with Springer. And many of you who are present there, like Prof. Samtapal, Prof. Lavanya, and many others, they are really leading this journal in best possible manner. So it's really great to be here and to deliver this oration. Now, Professor Amtapal ma'am asked me to talk about the genomic interventions. So before that, we need to understand why we need to do this thing. And as we know, being agricultural scientists, all of us are having several challenges, global challenges, facing food and agriculture. And one of them is about the population. And you will be seeing that continuously this population is growing. And very interestingly, it took the whole world for about 2000 years to reach 7 billion mark, and only 40 years to reach 2.8 billion. And if you see, majority of the population growth is happening in sub-Saharan Africa and Asia. And I was seeing today India's population, and just few hours back around 10:35, we are already 1.4 billion people. And you can see that we are having every day more than 34,000 births. If you see all around the world, right now, India is the second number, US is third number, and then in 2050, India is going to be number 1, China number 2, and we had some colleagues from Nigeria also when we started the presentation. Nigeria will be the third populous country. So now in this context, forget the future, even if you see the present, and this is the World Hunger Map of 2020, you can see majority of the countries, only few countries you can see in that these kind of mark in the blue and violet color, which are really good, and we need to have, in fact, we should have all the world painted in such color, but unfortunately, sub-Saharan Africa and also Asia, many countries here in Asia, you see this yellow color thing. This is not a good news, and several times even you are having these orange and then red colors. So this is not good. Right now, we are having about 821 million people chronically undernourished. And anticipation is that if this current trends continue by 2030, in next just 8 or 9 years, we will have this number reaching 840 million people. So hunger is a serious problem, and more than hunger, nutrition is also a serious issue. And if you see our country, India, more than 80% of the Indians, they suffer protein deficiency. Now, may I request all colleagues to do their mic mute so that we can have really good presentation. Anyway, so then protein remains a challenge for all of us.
Others can.
The unfortunate situation is that if you see the per capita consumption of pulses, that used to be 60 grams per day in 1950s, but this has been reduced to 38 grams per day. So we need to change the scenario. And of course, we can get this protein from a number of sources, and you can have the different sources, but then sometimes you are having the risk associated with the chronic diseases based on the source of the proteins. Based on these data, it seems that plant-based protein is really good because this is associated with the low risk of the chronic disease. Also, this is environmental sustainable. I will not go into the details about these things. And in view of that one, we at ICRISAT, together with our partners, many partners here in India and abroad in Sub-Saharan Africa, we have been working on 3 legume crops, chickpea, pigeon pea, and groundnut. And you can see that these legume crops, they are very important from the perspective of the nutrition, and also that to generate the revenues for the smallholder farmers. However, the crop productivity for these crops is not really good, and this is basically less than 1 ton per hectare in many of these countries. So now we need to work in this direction, and crop improvement programs around the world, this doesn't mean that nothing is happening, and many of these crop improvement programs and many CGR center, national partners, advanced research institute, they have been working tirelessly by using the crop breeding and allied disciplines, and they have been working on these different aspects, that how they can enhance the productivity, can address the issue of the disease resistance, pest resistance, can take care of the environmental stresses, sustainability, nutrition, etc. So a lot of things has happened. We got a lot of good varieties, but thing is that how we can enhance the precision and efficiency, and how we can enhance the pace for this crop improvement. And for that perspective, genomics technology can play a very important role. And in this presentation, I will be talking about that. Before that, I will be sharing some of our efforts and some background about the genomes and the genomics assisted breeding. So we all know the genome is basically a sum of all genes present in one organism or so. When we talk from the agriculture perspective, then idea is that how we can use the genome information, genes, for accelerating the breeding processes. So I will talk about that one. Now, genome is not a new term. We all know for many, many years, but basically that lot of interest started from 1990 when this International Human Genome Sequence Project was launched. And as a course of for about 13 years and the investment of $3 billion, they announced human genome sequence in 2003, and a lot of landmark work happened during this time. In fact, human genome was not finished completely, and even just 2-3 days back, many of you may have heard about or read in the Nature that basically just on the 4th June, researchers have completed now this human genome, and still they are having the 0.3% of error. What I want to say that the genome sequence is a continuous process, and this takes a lot of time to have the finished information. So now even the 2003, they announced, but now in the 2021, we have reached to this level. But anyway, so this was the case of that human genome sequencing or so. But let's come back to that agriculture. In 2000, we had the first plant genome sequencer of Rhodopsis thaliana. And I was in Germany when they announced this rice genome, and I used to work with Professor Andreas Graner at IPK Gutters level. And where we had these 2 genomes came out in 2002. And of course, at later 2005, we had the complete genome of International Rice Genome Sequencing projects. At the same time, we had a lot of explosion of the sequencing data in the form of the ESTs, and many genome sequencing projects started, etc. In 2003, I had an opportunity to participate in this conference from the Green Revolution to the Gene Revolution in 2003 in Bologna, organized by Professor Roberto Togrosa, where I met a large number of senior people, agricultural science stalwart, including Professor Norman Borlaug, Professor Swaminathan, Professor Gurdev Ghosh, Ronald Phillips, Professor Mike Gale, many people. And I think this conference was really very interesting conference, and this was talking that, well, how we can bring the gene revolution to enhance the crop productivity. In the same conference, I had really good interaction with Professor Norman Borlaug. And during his talk, and also after this talk, he mentioned, these words, they are still, I still remember those words. He said at that time, in the next 50 years, I challenge the next generation to use these new scientific tools and techniques to address the problems that plague the world's poor. And I think that this prompted me to work on the aspect that how earlier people were using the molecular markers, which is also one of the component of the genomics, but then how we can use the genomics technologies whole to enhance the crop improvement. And in that aspect, in the 10th anniversary issue of the Trends in Plant Science, they invited us, and together with Professor Andreas Graner and Professor Mark Sorrells, we gave this concept of the genomics assisted breeding for crop improvement, that how we can use the different kind of technologies. And then genomics assisted breeding kept on moving further. Few years later, with Professor Swapan Datta and many other colleagues, we also talked that how the genomics together with genetic engineering can be helpful in this aspect. And again, this was in the Trends in Plant Science. But during last 20 years or so, a lot of advances have happened in the genome sequencing. And I think in the month of February or March, these different journals of Nature, they came out with this milestone in genome sequencing initiative, where I was also one of those milestone advisors. And here they talk that how the genome sequencing have changed the landscape of the research, not only in the human, but in animals and plant sciences, etc. And now in the similar aspect, the genomics assisted breeding technology kept on changing the breeding strategies, that how the crop improvement should have happened. And just in the month of June, Trends in Plant Science, they came up with this 25th anniversary special issue. And in this issue, we also talk that how the genomics assisted breeding has come of the age, even in so-called minor crops as well. And now what is in future? And I will talk about this thing, and I talk this technology now, GAV 2.0. So far for last 15 years, we worked on the genomics assisted breeding. And now in next few years, we will be working on GAV 2.0, and I will talk about these things. So what I wanted to say that, well, a lot of things have happened during this last 20 years or so. And now in next few slides, I will be talking about these advances, that how things happen. And I think one of the objective of this talk is to inspire the young scientists, and then how they can take up the new approaches in their program, in their research. So let's talk about the genomics. So when we start the genomics, of course, you can study the different parts of the genome, but if you would like to understand the whole genome structure architecture, then you need to sequence the genome. And sequencing genome now, which has become much more easier, but I'm talking about 10 years back or so. So at that time, these kind of technology, they were just restricted to few labs or in few countries. But at ICRISAT in 2007, we initiated, or we established this center. We called it Center of Excellence in Genomics at that time. But in the 2017, this was rebranded as Center of Excellence in Genomics and Systems Biology, where my colleagues and my collaborators, they have been working tirelessly, and then they are generating a lot of resources for even so-called orphan crops or less studied crops. Many of you have visited. If not, we would like to invite you to join us, to visit us sometimes later, when we are having this corona situation calm down. Anyway, as a result of these efforts and our partners around the world, we have sequenced the genomes of pigeon pea, chickpea, pulse millet, and many of you know that groundnut, cultivated groundnut is a tetraploid. So we also have the two different genomes. So we sequence both of these genomes, and then two subspecies of groundnut, Festia jeta and Hypogaea. So we have led several of the genome sequencing consortium and understood the genome architecture, and we compiled all the genes. And this was a huge work, but we were successful to do this thing thanks to our partnership and collaboration. And we also had really good guidance from Professor Swapan Datta when he was the Deputy Director General of Crop Science at ICR in New Delhi. And we partnered with large number of organizations to achieve that one. Not only that, we have been collaborated or contributing co-lead the genome sequences of several other crops, including sesame, mung bean, ajoki bean, jatropha, and pea. We have just communicated this paper recently, longan, soybean, etc. So I think that we feel very happy to mention that our genomic center has taken the lead or contribute to sequence more than 10 different crop genomes. So this is really very exciting. When we started the sequencing the genomes, at that time point, technologies, the sequencing technology, especially next generation sequencing technology, they were their infancies. So for instance, Illumina sequencing technology, at that time, we used to have just 36 base pair reads. So we have assembled many genomes at that time point. But now with this power of the technology and a new technology called high-C analysis, where we are doing the 3D genome sequencing. So again, together with partners, we have improved these genomes. Now, for instance, in the case of chickpea, the earlier genome, we had the 8 pseudomolecules, but a lot of scaffolds. But now by improving these things, we have improved the chickpea genome. And this is that improvement that, for instance, that we have got the N50 from 39 mbvp to 65 mbvp. And genome was anchored also in the 92% genome was anchored to the 8 different chromosomes. In the case of pigeon pea, which was our first genome based on the just 36 base pair long reads, you can see the improvement was remarkable. And not only that, at that time point in earlier genome, we had the misassembled region, like for instance, chromosome 2. But now, by using the high-C analysis, we were able to split this chromosome 2 in the chromosome 2 and chromosome 5. And we also have found several sequences of the chromosome 9, etc. So now we have improved this genome and we are having the really good pseudomolecules. In the case of peanut, Arachis duranensis, Arachis hypanensis, also this high-C analysis were able to help us to improve these misassembled regions. So as I showed earlier about the human genome, that even after 20 years and despite of huge investment, the genome sequencing consortia, they are still working, scientists are still working on improvement. So this improvement work is in process, but we have not stopped utilization of the genome sequence for crop improvements, and we are continuing with this aspect. So once we got these improved genome assemblies, and I will talk something about basic science, and then I will come on the applied aspect. So when we got these different 6, so basically we improved 6 legume genomes by using the high-C analysis. And when we did the phylogenetic analysis based on all these different legumes, we found the 3 evolutionary event, which is eudicot common hexaploidy, which happened about 115 to 130 million years ago. The other event of legume common tetraploidy, which happened about 41 to 60 million years ago. And third, soybean specific tetraploidy, which happened around 5 to 18 million years ago. So based on these 3, basically these 3 evolutionary events have been responsible to make the different legumes from the common ancestors. And especially in the case of groundnut, there has always been a debate that how many millions back tetraploid peanut was synthesized from those 2 diploid progenitors. And our analysis also indicated that this is not very recent, because some group they say that, well, this happened just 15,000 years back. Some of our analysis mentioned that this happened much more earlier than this one. So again, there are several regions. And now when we got all these different legumes, we analyzed these different legumes together with Arabidopsis thaliana and rice to see that as an outgroup species. And then we try to identify that, well, what kind of gene families we are having common across the legumes. And here you can see that we got 516 legume family, which are present, gene families, which are present in all the legumes, but not in these other species. And then we did the geo-enrichment analysis, that what kind of genes are there. We found, which is not surprising, that the gene enrichment is basically of the nodulation, because nodulation are a specific phenomena in the legumes. Not only that one, because we had the groundnut part of that one, and groundnut or peanut is having the gravitropism, where after the emergence of this, when you are having the seedling and then this start to go towards that, towards this gravitropism. So we try to identify that, well, these are the genes that are involved in these legume specific phenomena, etc. And then further, we went deeper in these different legumes, and we found, and as I was mentioning, that this clade where we are having these peanut related peanuts, like this is tetraploid, this is the diploid. In this group, we had the enrichment or expansion of the gene families related to gravitropism. In soybean related thing, we had enrichment of the genes related to oil biosynthesis pathways, and species like chickpea, etc. We had that expansion of the gene families related to the photoperiodism, etc. So I think this work and all these things is still unpublished. So we have been working on these aspects. Sometimes some people say, hey, why you are working on those so-called orphan crops like palm millet or pigeon pea, how they are going to helpful to the food security in the world? Because people eat wheat, maize, rice, and they also do a lot of, if you see in the under the oil seed or legume crop, soybean is #1 crop around the world, or so, especially in the developed countries also. Now, answer to this question is that when we are talking about the climate change, and when we talk about the wheat, maize, rice, which were very, very important crops, but if you see the temperature sensitivity for these crops, pulp millet is the crop where you can have even the seed formation even at the temperature of 42 degrees Celsius. Many other cereal crops, they will not have the possibility even at the 35 degrees Celsius. So what we did, that when we had this pulp millet genome, we did this analysis together with the different genomes, and we found that at least there are 384 expanded gene families, which makes pulp millet so much drought and the heat tolerant, which is not present in the traditional or these so-called important cereal crops. So we are not saying that people start to eat wheat, maize, rice there, because I know that Mach and Bhat is very important in West Bengal. So this is not, we should not start to replace rice with the pulp millet. But what one can do, that by using these molecular biology approaches, you can transfer the genes responsible for heat and drought tolerance to improve the rice, so that you can have the higher productivity in this heat and the drought stress condition. The another interesting example of pigeon pea, and this pigeon pea, which is called an orphan legume crop. Many people around the world, they don't know about pigeon pea, but people know about the soybean. And in the case of soybean, there is an issue which is called that basically Asian soybean rust, and there is no natural variation for Asian soybean rust. But when we got this pulp millet genome sequence, a group led by Jonathan Jones from the Sainsbury laboratory, he talked to us and we provided the genome sequence of pigeon pea even before the publication. And the soybean group was successful to identify a gene from pigeon pea, which confers the full resistance to Asian soybean rust. And here you can see that one. So basically what I'm trying to say, that this kind of translational approach, that you can translate the information from pigeon pea. Just imagine if we wouldn't have the pigeon pea genome sequence, you wouldn't identify this particular gene. Here in the case of soybean, you will not have the possibility to improve this thing in the faster manner. So this is the way that we should not be worried that this my crop species is orphan crop or minor crop or something. Hey, we need to work on our crop, and we need to provide these resources in the public domain so that the other crop community can also make the best use of these resources. Now, friends, I was talking about the genome sequences. So genome sequence can provide us that information about the genes. Like for instance, in the case of chickpea, we got 28,000 genes. But the question is, are these 28,000 genes, they are expressed in all the tissues at all the times? No, this is not the case. So then to understand that at what time point, which gene is expressed in which tissue, in which condition, we have assembled the gene expression atlas, and we developed the gene expression atlas in the case of chickpea. Now we are using the gene expression atlas to understand the mechanism for the drought tolerance in the case of chickpea. Same thing, we have developed this gene expression atlas in pigeon pea. In pigeon pea, we are having the hybrid breeding system, and now we are using this system, or we are using this gene expression atlas of pigeon pea to understand the mechanism of cytoplasmic male sterility, and we already got some really good leads. This work was also published last year in the journal, the Plant Genome Journal. I haven't forgot to mention the journal name here, but anyway. And the same thing, we have developed the gene expression atlas for groundnut as well. And here you can see some important phenomena, as I was mentioning about this gravitropism, etc. earlier. So anyway, so we got the gene expression atlas. So I was talking earlier about the genome sequence, then I'm talking about now the gene expression atlas. Now, question is, and nowadays all world is going about the pangenomics. And what is the pangenomics? So what you do, that when you sequence one individual of any species, you identify the gene, you collect, get the information of genome of that particular individual. But does that mean that all other individual of that particular species will have the same set of the genes? Recent research has mentioned that, well, this is not the case, and sequencing of single genome is not enough. And now based on the sequence, and so for instance, here, this picture, you are got this 3 cultivars, 1, 2, 3. Now, when you sequence these information, then they have found that when you compare these things, that some sequence, this is common across all 3, some sequence is common between 2 and this between 2, and some is still unique to each of these cultivar. Who knows that some of the interesting traits in which you are interested, they are present in cultivar 3, they are not present in cultivar 2 and cultivar 1. So basically we need to assemble the genome for the cultivar 3 as well. And here the gene content which is common across all the different individuals called core, and which is common across the between 2 or so, this is called dispensable genomes or so. So people came up with this concept of the pangenome. This came from the bacterial genome sequences, and now many crop community, they are moving in this direction. And then our group, we also came up with the new concept called super pangenome, and we say that this pangenome concept should not be restricted to only at or to one species level. This should be extended to the genus level. So what is happening? So for instance, in the case of chickpea, Cicer is the genus, and here you got the 8 different species. So what we are telling that now, for instance, Cicer reticulatum, and here you got a lot of individuals. So by using these lot of different individuals, you sequence them, develop a pangenome for Cicer reticulatum, develop a pangenome for Cicer cuneatum, develop a pangenome for Cicer gyratum. So like those kind of thing, develop a pangenome for Cicer amaceti, and then after that, combine all these different species pangenome and go at that genus level, and we call this thing the super pangenome. And with this concept, what we started to do, we have sequenced the genomes of all the 8 different wild species of Cicer. And I don't have much time, but I will like to go a little bit faster. So then what we are doing that now we are comparing those 8 different genomes with the Cicer reticulate, Cicer hereditatum, the cultivated one. And then after that, we are going for this species level pangenome. And when we have done this analysis of all the different species, we found that there are only 50% of the genes that are present as a core, and the remaining set, this is the dispensable. And then you can also identify the species specific genes and can understand the mechanism about these different genes further. And not only that one, then when we did the pangenome modeling, so what happens then, the modeling means that when you are having the number of exons, number of the genomes, and you are having this number of the genes, so by using these different modeling, we have identified, so what happens that when you keep on increasing this number of the genomes, you can see that after when you increase the number of genomes, the number of genes keeps on increasing, but after some time this becomes like plateau. And this kind of thing is called that this thing is that closed pangenome, and we are trying to identify this thing. So this was that basically idea that sequencing one genome or one species is not enough, and you need to continue to sequence large scale exosomes. This is much more important when you are having the huge diversity. So for instance, these are the chickpea, and these are the different seeds. You can see the different kind of colors, shapes. I'm not talking about the traits like disease resistance, drought tolerance, etc. And of course, these different exosomes will have the different level of resistance tolerance. But here you can see from this color as well, from the shape as well. So each of these exosomes, they vary from other. And now to understand this thing, we need to sequence the genomes. So what we have done that in the case of pigeon pea, we sequenced in the first instance about 300 lines, 292 pigeon pea exosomes coming out from the 23 different countries. And this information was very helpful to understand the center of origin of pigeon pea in Madhya Pradesh, and also try to identify the genes related to with different agronomic traits, etc. And same thing in the case of chickpea, where we sequenced this chickpea with more than 300 lines from 33 countries. So here we are sequencing the entire genomes, and then again we are doing a lot of analysis. I will come back to this point later. But one thing in the case of chickpea, for instance, many of these crops they had the center of origin in the Mediterranean regions, Fertile Crescent, and this sequence data also provided us the information that how they were migrated to the Middle East, and then after that how this moved to India and the parallel to Ethiopia, and then further this moved to the other parts of the world like in United States and the Australia or so. So this is the way that you can get a lot of information. And after that, we have sequenced now recently more than 3,366 genomes from all the over the world. And now we are still analyzing these data. But one thing I can mention that earlier we are having the pangenome based on the species. Now we got the pangenome based on even the cultivated exosomes. And here you can see that when you are increasing this number further, the genes number is not going further higher. So again, this kind of information will be very, very helpful. We also use this analysis to understand the center of origin and also the center of the diversity. And after this analysis has provided that there are 4 centers of diversity of chickpea, which is in the Middle East, Mediterranean regions, Central Asia, and South Asia. And yeah, so when you get these kind of sequence information, variation, etc., the next thing is from the breeding aspect, we would like to use this information in the breeding program. And for the breeding program, what we need to have either genes associated with the trait or molecular markers, and they should be on some platform so that they can be used to screen the breeding population. For that purpose, we have developed a range of the genotyping platform, marker genotyping platform, and our approach has always been that how we can make these genotyping platform the cost effective. And now in each of our crops, we have developed the SSR genotyping platform, DART, CASPR, genotyping by sequencing, whole genome resequencing. We also developed the SNP array, which is having 56,000 SNPs in one array. So you just take one experiment, you get the data for 56,000 SNPs. We got 2,000 SNP array, which can be used in the genomic selection. We got the 10 SNP panel, which can be used in the marker assisted selection. So we got a range of the marker genotyping platform. And here I'm giving just an example of the case of chickpea, for instance, 10 SNP panel. This you can screen in just $2, one line, including DNA extraction. Our colleagues and collaborators are using this panel for molecular breeding for drought tolerance, Fujiram wilt, Ascochyta. Earlier, this process was very expensive. Still, many labs, they're spending more than $10 to $20 per sample. We brought this cost down to $2 per sample. Now, in the breeding, we need to have the information about the purity of the parents. And then we have developed these QC panels. We also developed the SNP array where you can have the 50,000 SNP in just $55 per sample. These panels, they have been used to map several traits. Recently, the Plant Genome also put this cover article where we have mapped the helicorpa resistance component traits in the case of chickpea. Now, my colleagues and collaborators, they're moving towards deploying the genomic selection. For that, we don't need to spend $55 per sample. And here, we have developed the platform where we are having 5,000 SNP and they can be used in just $6 per sample. So my friends, we have developed these different kind of platforms. Now, as I mentioned, you need to have the trait associated markers. So for that, we need to use the range of these populations. And this picture is coming from my colleague Abhishek Vora from IIPR Kanpur. And then what we are doing that we are having a different kind of biparental population, multi-parents population, association mapping population. And not only that one, we are using the next generation trait mapping approaches where instead of screening the entire population, we can just sequence just the pools of the population. So I will not go in the details. If you are interested, send an email or get the screenshot of these slides and keep on reading the papers. But what I want to tell that we have used a range of the fast forward trait mapping approaches, linkage mapping approaches, association mapping approaches. And based on these things, we have mapped from 20 to 50 traits in each of these crops, chickpea, groundnut, and pigeon peas. So I think this was a huge work. A lot of collaborators work, a lot of colleagues from ICRISAT and our national partners, but we have reached to the level where we can now deploy the molecular breeding. Earlier, these crops used to be called orphan crops. And when I joined ICRISAT way back in 2005, when I used to talk to the people, they used to say, hey, you are talking about the genomics assisted breeding in these crops where even you don't have the genetic maps. But friends, in the partnership, in the collaboration, and with the power of the technology through the consortium, we can deliver these kind of things. As here is an example that in these three crops where we did not have anything, now in starting from the genome sequence, now we got the sequences for thousands of the exosomes. We got the marker associated with the different traits. So we have reached to this level. Now, the next component is that how we are translating this information from the genome to the field. And here, this is the picture from ICRISAT from the groundnut field. Here, you can also see from your eyes that whenever we are having these kind of, you are having these healthy crop and the disease resistance. So these disease resistance lines have been developed through marker introgression. And here you can see sometimes some check and sometimes some of these, basically the elite parents, which we improve. So anyway, so now by using these different kind of approaches, and now we come back these genomics assisted breeding approaches. In our earlier paper in 2005, we mentioned different approaches, including marker assisted selection, marker assisted backcrossing, and also ABQTL approaches, many approaches, and some approaches they came during last 10-15 years like Mars or genomic selection, etc. But nevertheless, during last 15 years, these approaches have been used very extensively in the context of the genomics assisted breeding. Now, what is going to happen for the 2.0? And we came up with this idea with that new approaches like haplotype based breeding, genomic selection, and also optimal contribution selection. Another gene editing approach, and I will talk that many times people are using gene editing for improving the gene. You can also use them in the breeding program. And then some of my collaborators from Edinburgh Roslin Institute, we have discussed these things in some of our earlier paper. We think that you can promote the superior alleles or you can remove the bad alleles through the gene editing, and this approach called PAGE or RAGE. So anyway, so now, and then these approaches now they can be combined together with the speed breeding, and this concept came from Lee Hickey from University of Queensland. Now many institutes around the world, they're integrating this thing, including ICRISAT. My colleagues are establishing the facilities of speed breeding at ICRISAT. So anyway, so now we will discuss some success stories from GAV 1.0, and now we will discuss some potential from the GAV 2.0, that how we have been successful to use the genomic interventions in the cow breeding. I don't have time to go in the method parts. I will just tell the outputs. And here I feel so excited that together with our partners, we have been successful to deliver several improved varieties, because people still keep on thinking, hey, this biotechnology, they just are the promising technology, they do not deliver. This is wrong. Many biotech approaches already delivered. Here I'm talking some examples. Now, we had this QTL hotspot from the drought tolerance, and we have introgressed this QTL hotspot in the JG11 background, and which was responsible for higher root biomass, etc. You can see many lines that they are having much more better root biomass, root length, etc. than JG11. This was the cover article few years back. But the good thing is that one of this line, after multi-location evaluation, was released in Ethiopia, and this was the first product of chickpea for the drought tolerance through molecular breeding, genomics assisted breeding, and this variety was named as Geletu in the honor of one of the Ethiopian plant breeder, and who did a lot of work. So I think that this variety is already reached to the farmers now. Second, from our country itself, from here, and this breeder, Dr. Bhardwaj, fantastic breeder, and he and we have been working for many years together. And now one of this variety in the genetic background of Pusa 372 was also released as a molecular breeding variety for the drought tolerance with higher yield by Government of India, and this work was in collaboration with Indian Agriculture Research Institute. The other product coming from this molecular breeding for the Fujian wilt. This breeder, Dr. Mannoor, and he was a breeder in the University of Agricultural Sciences, Raichur. He's retired now, and his colleagues Yeri and also Lakshman, and we worked, and he worked together, and we developed some of these lines. And one of this variety basically, so Super Annigiri. This Annigiri was is still very popular variety, but became susceptible for Fujian wilt. And we have developed a new line which he calls Super Annigiri, and this was also released in India 2 years back or so. And now another variety, this is also going in Madhya Pradesh and other states also. And recently this year, we got another variety coming from IRI from Dr. Bhardwaj, and this came, and this was named as a Pusa Chickpea Manav for the enhanced Fujian wilt resistance. And all these things are coming through the genomics assisted breeding efforts, which I mentioned few slides back. And this was fantastic work. And this kind of work gives me much more satisfaction than the genome sequencing papers in the Nature. Why? Because here, this work is a collaborative efforts of several institutes of IRI, IIPR, and ICR organization, State Agriculture University, and funding from the Ministry of Agriculture, and of course, collaborators from ICRISAT. You can see this work reports the introgression of the QTL hotspot in 3 elite varieties. And in each of these cases, this has enhanced the drought tolerance. In many cases, these lines have already reached, in addition to the previous one, they're in the ACRIP trial. So I think this kind of the translational work is really good. And this kind of thing has also been highlighted by several organizations like Crop Science Society of America, American Society of Agronomy, etc., breeding a better chickpea. So we are doing this thing here in India, but work is being popular even in United States, that how this group was successful to deliver these kind of products. Here, even here in India, some newspaper, they are talking about the genomics behind the science of making India super chana. So I think these things bring a lot of satisfaction for us. Similar kind of efforts are underway in the Indian Institute of Pulses Research, where we are working with them. They are also improving for Fujian wilt resistance, etc. Same thing in the case of pigeon pea. Many universities in different states, we are working to improve the Fujian wilt and SMD in the case of pigeon pea. Now, another success story I would like to mention here in the case of groundnut. Many of you who know about this groundnut crop, then in that groundnut is an oil seed crop. And if you see the oil, then this oil is having basically 3 different components: palmitic acid, linoleic acid, and oleic acid. Linoleic acid is not good for health. Some American varieties, they are having more than 95% or 90% oleic acid. Now, by using the genomics assisted breeding, together with DGR of ICR, we developed several lines which are having more than 80 to 90% oleic acid. And Government of India, together with DGR, they released 2 varieties, Girnar 4 and Girnar 5. They were the first set of the high oleic groundnut varieties. So, and not only that, one Prime Minister Modi last year, when he dedicated these biofortified crop varieties to the nation, then these Girnar 4 and Girnar 5, they were also part of that one. So I think that now this kind of work is moving further. Now some of our collaborators from US, Dharwad, they're moving ahead. And in the case of groundnut, we have also improved that lines for the foliar disease resistance. And now these products that's also been transferred to several countries in the West and Central Africa. This picture is coming from Ghana, where I was visiting some field, and some of these lines, they're being coming up very nicely, which we developed through the molecular breeding approaches. Now we are combining the high oleic plus foliar disease resistance together. And again, here you can see in this cover article that here you got this susceptible line, here you got this resistant line, and these lines, they're coming up again by combining these things together. So I think that a lot of good things have happened, are happening, and not by ICRISAT. And ICRISAT believes in empowering and collaborating the partners, that our partners should be doing this thing. And I feel so happy that now even like in Ethiopia, EIR, in Kenya, Egerton Agricultural University, and the same thing in India and many other countries, they are taking this genomics assisted breeding legacy further, and they themselves are using this thing. Now, as I mentioned, and I think very briefly, I will talk about this GAV version 2.0, that what is this genomics assisted breeding 2.0? Again, together with several colleagues, we have come up with this concept and idea. Now, because of the sequencing technology, it is possible to sequence the entire gene bank. If not entire, a sizable portion of the gene bank. And once you got this sequence data, you can identify 2 things. One, genes associated with the trait. For that, you need to combine these things with the phenotyping. Second is the superior lines, which are having combination of several genes, the good genes. Now, if you would like to take in the breeding program, this has always been an issue in the breeding program, that if you would like to bring the diversity, then sometimes you bring the negative traits, the linkage drag. So basically, you need to have this balance, that how you should have higher genetic diversity, but at the same time, the good traits, not that when you are bringing the good traits, then you don't have the genetic diversity. If you go to the genetic diversity, you don't have the genetic gain. So this is really, we need to do the balancing. And now we can do this balancing because we got the massive genome information. Now we need people to harness, to utilize the genome information. So this is point number 1. And I will talk about this thing, that we can do this thing by using the approaches like optimal contribution selection, that how you will like to select the parents which are having the traits, the diversity, but at the same time, they are having minimum linkage drag, and this approach called optimal contribution selection. Now, the second approach is that when you get the genes associated with the trait, you go further down and further in the details and then identify the haplotypes. I will discuss about that one. And then you move towards the haplotype based breeding. Third point is that once you are having the genes, you do the functional validation by using the approaches like systems biology approaches. And then once you got these precise genes, then you do the gene editing and you can move ahead. Further, you also use this information for the genomic prediction. And lastly, once you are having the sequencing data, you can also identify the genes which are having the deleterious effect, and we call them genetic loads. And you can also use that editing. So basically, you need to use the genetic editing, both gene editing for both purpose, for beneficial alleles as well as for the harmful alleles. Nowadays, what we need to do, we do not need to just focus ourselves on beneficial alleles, and we have been doing great job in past. My suggestion is that we should also now start to work to purge these harmful alleles either through traditional breeding, through genomics assisted breeding, or through gene editing. And we can do this thing because we got a lot of information, and we can, by using these approaches, we can really develop the future crops. Now, again, you will find a lot of information in these published articles, but very quickly. So when you have the gene, and we have been understanding earlier for the alleles. So for instance, you got this one gene and you can get these different alleles, you have got the other genes like that one. And the combination of the different alleles for a given gene is called haplotype. And here you can say that, well, this is the haplotype H1, this is haplotype H2, this is haplotype H3, like that one. Now, once you are having the sequencing data for large number of genotypes, you mine those genotypes for these haplotypes, and then you come up with the idea, oh, this genotype 1 is having haplotype H1 for gene 1, haplotype H1 for gene 2, haplotype H2 for gene 3, like that one. So you get this cataloging of the different haplotypes. Now you come to the third point. In the case, for some of these lines, you got the phenotyping data. That's great. If not, then generate the phenotyping data and then do the analysis. We called it haplopheno analysis. And when you combine, they do this haplopheno analysis, then based on these things, you identify. So for instance, in the gene 1, the haplopheno analysis indicates H3 haplotype is better. Here for gene 2, this indicates that for root depth, for instance, H1 haplotype is better. This says that H2 haplotype is better. So basically, based on this haplopheno analysis, you identify the superior haplotypes. And then you go back to your elite variety, ask a question that what kind of haplotype you have for gene 1. For instance, this says H2, but then we realize, oh, for gene 1, H3 is a better haplotype. So now we should replace this H2 haplotype by H3. Same thing, we need to think, what about for gene 3? Oh, gene 3, this elite variety is having H4 haplotype, but I found from my analysis for gene 3, H2 haplotype is better. So how you can bring? So basically, our idea is to replace these non-desirable haplotypes by the desirable haplotypes, and this approach is called haplotype-based breeding. At ICRISAT, we already started to work in this aspect, and now, for instance, that we did this work for the pigeon pea. Some of you, I know that Johiruddin Molla came from Calcutta University, Professor Sopan Datta recommended, but he went back now. He was also involved in this part of that work. And what we did that, for instance, for drought tolerance related genes in the case of pigeon pea, we identified the haplotypes. So for instance, for plant weight, for fresh weight, for the turgid weight, we identify that one. For instance, this haplotype, and this haplotype is good. And then we are asking the questions like these improved, this ruling variety like Maruti, which is a ruling variety in Karnataka and other places. Now, if you say this is ruling, but then this got several inferior haplotype. Now, if you can replace this inferior haplotype with the superior haplotype, you can make Maruti as a super Maruti. And that's our job, that how we can do it. So I think that this is the way now to move ahead about the haplotype-based breeding. Second, I will talk about the genomic prediction, which is coming up with very powerful, in very strong manner. So what is happening? If you see the animal breeding and the plant breeding, in plant breeding, majority of time, we are giving a lot of emphasis on the product development, that how we can develop superior variety. In the case of animal breeding, they talk more about the population improvement, that how the next generation of their breeds, the animals will be having will carry much more beneficial traits. So basically, they can keep on doing this thing there. Traditionally, the breeding program, they were talking a lot about product development, but now this is the time that we need to start working more about the population improvement. We need to have sequencing, genotyping of each generation. We need to predict these things and we need to take them in the next generation of the crosses. And this is the way we can move ahead. Many people still believe that genomic selection cannot happen, this, not that, etc. Well, we need to be the optimistic, we need to be ambitious now in I think across all the different crops, including our crops. Now there is a possibility, we got the genomic resources, we got the databases, we got the genomic selection pipeline, we got the phenotyping platform, we are developing the capacity. So by doing these things, it is possible now to select the lines. And again, you can do the genomic selection for both things. One, which line is having higher breeding value, they need to be taken to the next generation. Second, which lines are not good and can you discard them at early generation itself. So basically combining these things together, we can do the genomic prediction. Again, together with IRI, IIPR, Cornell University and Bose and our colleagues are leading this work. So for instance, this is coming from my colleague Manish Rudkiwal, and he's leading this project now in the genomic enabled prediction for the, in the case of chickpea, for instance. And by working on this aspect, we have identified some traits, some models where we can have up to 0.91% of prediction efficiency. And then we went further for using the multi-environment trials, because you cannot believe in one is location data. I know I cannot go into all the detail, but I just would like to give you the flavor, and who are interested, they can read these papers that how you can enhance this prediction accuracy further. Similarly, another colleague of mine, Manish Pandey, and he's leading this project on the genomic-based trend prediction in the groundnut, and together with director of groundnut research, etc. And then we have come up with this idea about enhancing the prediction accuracy up to 0.6. And similarly, we are working again in the case of pigeon peas coming from Rajeev Saxena and other colleagues from IIPR, ICRISAT or so. And here, what we have done, we worked this thing on the IPK gutters level also. So now we have done the whole genome sequencing of the hybrid parental lines. And by doing these prediction, we have also able to identify the best parents, which were not used in past for making the crosses. But if you will use those crosses, then they can deliver much more better hybrids. And some of these combinations are being tested by Dr. Abhishek Bora from IIPR, and we hope that these kind of approaches will be much more helpful. So I think this was the genomic prediction and how you can enhance this thing. In fact, the 3000 genome sequencing project, which I was talking earlier, has also provided us 19.10 million hapto-pheno combination, and you can screen a large number of superior haplotypes. But for few traits, we have done detailed analysis and we found the superior haplotypes for the breeding yield related traits, and now we got this information, we can transfer these things in the lead varieties. Same thing for the genomic prediction, not just by using, as I told, the optimal contribution selection. So like for instance, that these are the candidates, the red color thing, this is the green color progeny or so, and then how you can enhance in the different areas. And this is basically Bayesian generalized linear regression. This is the HOGEM approach, haplotype based local GBV. So by using these information, you can also enhance the genomic prediction. Friends, many of you are already working in gene editing, so I will not go into detail. This is another important approach, but for gene editing nowadays, CRISPR-Cas is very, very popular. But one thing is that for these kind of approaches, you need to have the real gene, the gene which cause the effect. So causal gene, which are responsible for phenotype. And for that, for instance, International Rice Research Institute and some German institute and United States, they did fantastic work for genome editing for the bacterial blight. And few years back, we were also asked by Nature Biotech to write a commentary. And here, one of the messages that I did a lot of genome information, genome sequencing on the 3000 rice project, they came up with these candidate genes, and then only they were able to stack them. So basically, to understand, to have the product through the gene editing, you need to have the gene. So this is very, very important. And now to identify that particular gene, many times people are doing transcriptome analysis, etc. In my opinion, this is not the way to move ahead. Now you have a possibility to generate the data. First is that we should generate the data by asking biological question and hypothesis, not just like that. Second is you have a possibility to generate the genomics data, transcriptomics data, proteomics data, metabolomics data. Idea is how you can combine these things together, integrate this thing, use the network biology approach, modeling and simulation, and based on these things, you come up with the gene which are linking the genotype to phenotype, and then only you use the genome editing approach. So this was one of those approaches that how you can do it. Second is, as I told earlier, not only for the superior genes, our 3000 genome sequencing project also has provided us the information about the genetic loads in chickpea. What does it mean that this genome is having the poor alleles, the harmful alleles, the deleterious alleles? So the question is that how we can address. So if you would like to use them, then you can remove these alleles from those things, and we call the removal of alleles through the gene editing. So basically, you can use these approaches. And another last important, very important thing, that breeding, we need to accelerate, we need to enhance this process. Generally, the breeding cycle takes 6 to 7 years. If you are having one cycle takes 1 year. So this is the traditional breeding, will take 7 years to reach F6. Shuttle breeding, few years back, well, not few years, several years back, concept from Norman Borlaug, and he gave this concept that you can do the 2 generations in 1 year by shifting the location. This was also helpful, but now Lee Hickey and his colleagues have come up with this idea that you can do even 6 generations 1 year. So you see that although genomic breeding approaches, what I was talking, haplotype based breeding, genomic prediction, optimized contribution selection, etc., if you combine those approaches together with the speed breeding, you can do wonders. So basically you are putting the genome information and you are doing these things together. So in summary, where we are, and I think I can also highlight 2 examples, because recently, as I told, that Nature Biotechnology also had the 25th anniversary. Trends in Plant Science, they also had the 25th anniversary. And we were asked by both of these journals that where the crop breeding will be going, how this thing. So they were talking that how the future is expected to happen. I will not go in the detail, but in the Nature Biotechnology, we mentioned that in coming years, we anticipate generation of massive amount of sequence data, not only at the whole genome level, but even the single cell level, not only one line, but even the population level. So what is the next we need to do? We need to use the artificial intelligence and machine learning languages, that how we can use these data set to enhance the accuracy of the genomic prediction. Similar kind of thing we mentioned in this Trends in Plant Science, and we told that, well, nowadays, as I said, that like I was mentioning earlier people were using the normal approaches, but we believe the system biology approach is going to be norm for understanding trait biology. When we got all these information, we will be able to breed the new desired genomes based on the rich information, what we are generating. We believe again, this impact of this, basically this huge sequencing data needs to be used together with the phenotyping, and by leveraging the powerful data science tools. Again and again, we believe that by having all these things together, we should be able to have much more better prediction of the lines which needs to be used in the crossing program. And we are very much hopeful to see that plant breeding community will be armed not only with the vast area of the data, but also the proper tools and technology to decipher and implement the knowledge to feed 10 billion people. And the slide from which I started earlier, that how we can do it. So I think we are still very hopeful we should be able to deliver. Now the take home message from this presentation is, and as I try to demonstrate, the genomic innovations, they can enhance the precision and efficiency of crop improvement program. Translational genomics has come of the age even in so-called orphan or non-model crops. I already showed the delivery of several products. They are reaching to the farmer's field now. Now, genomics assisted breeding 2.0 is coming up with the new set of the approaches, including haplotype-based breeding, genomic selection, gene editing, along with the speed breeding, and they are going to accelerate varietal development process. And I hope in coming years, we will be having the combination of the artificial intelligence and machine learning in these approaches as well. Friends, I talked one component to deliver the better varieties. As an agricultural scientist, we know that just delivery the variety is not going to solve all the problem. First, they need to reach to the farmers in the real time. Second, if you really would like to harness the potential, the genetic potential of those varieties, you need to take care of agronomy, you need to take care of the management, and then you will have the higher produce. And if you got the better produce or higher produce in the farmer's field, you need to connect farmers to the market so that they can also reap the benefits of these produce, so they can have the better price. And for doing these things, we need to have the international, national, and local government agency support. We need to have the conducive policy environment when we are talking about the approaches like gene editing, etc. So I think that's what I wanted to mention. And here in the end, I would like to thank you all for all the partners, donors, and supporters, and all of my colleagues from the Center of Excellence in Genomics and Systems Biology, my collaborators from ICRISAT, my collaborators from more than 35 countries with whom I really had a privilege to work with them. And once again, I would like to thank Professor Archana Sharma Foundation, and especially Professor Amtapal for giving me this opportunity to share this work. Thank you very much, and I would be very happy to take up any question if you have. Thanks a lot for your time. Madam,
thank you so. Yeah, thank you so much, Dr. Varshney. We wish it was a series of lectures, because you took us from conventional plant breeding right through the realm, through all the innovations that you have contributed so much into, and right to the realm of artificial intelligence and machine learning. So I really wish, as a teacher, I wish that my students be exposed to the wealth of information and the wealth of your research acumen. And I would like make a request to you that when things go back to normal, if you can share this, like your wealth of experience with our students in a series of lectures or something, like given your time and however your convenience works out. But it is really amazing. I mean, all of us, like there have been so many comments in the
Yes.
chat. So many people are saying that, like, it was obviously, it goes beyond saying actually that what you had, the journey that you had taken us from, it's really amazing. And the future that you have shown us, it's really, really intriguing. As Professor Varshne has indicated, he can take a few questions. So if you can please write it in the chat box, I can field a few questions to him and he can answer that.
Yes.
So there are already a few questions, Professor Varshne. I'll start with that. So Parmeshwar Sahu is asking, he has a basic question related to association mapping. He's saying in GWAS, we identify markers which are associated with desired traits. And since there are such a huge number of diverse germplasm lines as experimental materials, he wants to know that how can you identify these genotypes which have the major alleles of linked markers, or how we can identify those genotypes which have the genes we have identified through GWAS.
Yeah. Thank you, Dr. Momita, for very nice moderation, and thanks to all the colleagues who have put this appreciation and that good remarks in that chat box. I can read many of them, I can see. And of course, I will try to answer several of these questions, but I think that there will be limited time. But those of you who are not able to get the answer, please feel free to send an email or send a tweet message or something. We'll be happy to do it. Now, coming back to this question of Parmeshwar Sahu. Yes, so when you use the GWAS, there is always the issue that you are using the diverse set, and sometimes the markers or the genes which you identify, they may not be very valid in that elite germplasm. So this is really very important that you need to do the validation on elite germplasm. And to address this issue, what we have proposed that once you are having the GWAS, especially in the species where you got the genome sequence, you identify the genes, and then based on these genes, you go in the process of identifying the haplotypes, go through the set of the elite lines, and then based on the phenotyping data, then you ask a question that the gene or the haplotype which identified through GWAS, how good it is, and if you find any lines which is having the superior haplotype, use that in breeding program and also use the markers associated with this haplotype, and then you can move ahead.
Thank you. I see Dr. Soumitra Pal, my colleague, he's raised a hand. Soumitra, do you have a question?
Yes, thank you, Professor Varshne. This is Soumitra from University of Calcutta. So thank you for your nice presentation. One of the important questions that what is any initiative has been taken by the government of India for the Lathyrus genome sequencing, because Lathyrus being an orphan crop, it is much needed in our society. So
thank you. Yeah, thank you, Dr. Soumitra Paul. I don't have much information, but I know that some people from John Innes Center, and they contacted Indian Institute of Pulses Research also. At one time point, I was also involved in those discussions. So I believe that John Innes Center in UK is taking the lead. And I think in coming, so you see that genome sequence is not a big issue now. And even our laboratory can sequence the genome. If you are interested, we can work on that thing. But thing is that we need to have some biological questions. And as you said, then in Lathyrus, we already have these things. And I think that we need to add it, we need to fix those genes which are not useful for health. So I think
low ODAP content is very much necessary. And there are some varieties although, but I think genome editing can help to develop some low edited varieties in future.
So I appreciate that, and I think I strongly support you in that aspect that we need to work in this direction. Thank you.
Okay, thank you, sir.
So there are lots of questions. Probably we'll run out of time. And as Professor Varshneey said, you can all contact him directly. He will be very happy to field all your questions. So keeping the constraint of time in mind, I will now request Professor Rita Kundu, who is the Secretary of the Archana Sharma Foundation, for the conventional vote of thanks. So just before Rita Di comes in, I would like to mention one thing, that as a part of our oration program, we actually hand out a memento, a plaque as a memento to the speaker. This time, due to organizing it on an online mode, we really regret not having the opportunity to hand it over to Professor Varshneey during the program, but we will be posting it to you as soon as we can. And we still retain the hope that one day you come to Calcutta University and we have a face-to-face meeting and our discourse continues.
Thank you very much, Momita. And before Madam starts, I would like to thank you all once again, and would look forward to receive memento. Thank you for this honor, and also thanks to all the audience and for their kind words. So I really feel very excited, and thanks once again to all the organizers. Thank you.
Thank you, Professor Rita Kundu.
Thank you, Momita. On behalf of the foundation, I would like to thank our honorable speaker, Dr. Rajeev Varshney, for his wonderful lecture. Thank you, Dr. Varshney, for your amazing and mesmerizing presentation. Despite your busy schedule, you have agreed to deliver the lecture, and we are thankful for that. I must thank our head of the Department of Botany, Professor Binoy Chaubey, for his assistance and encouragement. I also like to express my sincere thanks and gratitude to all my colleagues and faculty members and the participants who are attending this program online. I would like to thank Mr. Akash Mundul of Web Wonders for his technical support to hold the event successfully. Last but not the least, I would also like to thank Dr. Momita Bandyopadhyay for conducting the event very smoothly. And I also want to thank our President, Professor Amita Pal, for arranging this beautiful lecture from this foundation. Thank you very much. So now I request Dr. Momita Bandyopadhyay.
So without much ado, we come to the end of the session, which has been very fruitful for all of us. I know I speak on behalf of everyone who has attended this session. Again, a heartfelt thanks to Professor Amita Pal for arranging this wonderful lecture, and Professor Varshneey, thank you so much for sharing your experience and your excellent topic of discussion with us. Hoping all of you have a very safe and happy time to come. Thank you very much from Archana Sharma Foundation of Calcutta. Thank you very much.
Thank you very much once again. Have a nice day.
Thank you. Same to you.
Thank you.
Thank you. Thank you so much. Have a nice day.
Thank you.
Thank you, Mr. Momita. Thank you, Professor Varshneey.
Thank you, sir, for a very nice presentation.
Very well done.
Thank you, sir. Thank you, sir.
वो आया क्या?
Thank you, Mr. Mondal. This went seamlessly, actually. Thank you so much.
Thank you, ma'am. It was great. It was great. Yes. We learned a lot also hearing this.
Yeah. So plant biology is now going into artificial learning, right?
Yes.
Artificial intelligence and machine learning. So how's that?
Thank you so much, ma'am. Thank you.
Thank you.
Thank you.